Chapter number 5, Principles of Inheritance and Variation. Have you ever wondered why an elephant always gives birth only to a baby elephant and not some other animal? Or why mango seed forms only a mango plant and not any other plant? Given that they do, are the offspring identical to their parents? Or do they show differences in some of their characteristics? Have you ever wondered why siblings sometimes look so similar to each other or sometimes even so different? These are several related questions are dealt with scientifically in a branch of biology known as genetics. This subject deals with the inheritance as well as the variation of characters from parent to offspring. Inheritance is the process by which characters are passed on from parent to progeny. It is the basis of heredity. Variation is the degree by which progeny differ from their parents. Humans knew from as early as 8000 to 1000 BC that one of the causes of variation was hidden in sexual reproduction. They exploited the variations that were naturally present in the wild populations of plants and animals to selectively breed and select for organisms that possess desirable characters. For example, through artificial selection and domestication from ancestral wild cows, we have well-known Indian breeds, that is Sahiwal cows in Punjab. We must, however, recognize that though our ancestors knew about the inheritance of characters and variation, they had very little idea about the scientific basis of this phenomena. 5.1 Mendel's Law of Inheritance. It was during the mid-19th century that headway was made in the understanding of inheritance. Gregor Mendel conducted hybridization experiment on garden peas for 7 years and proposed the law of inheritance in living organisms. During Mendel's investigations into inheritance pattern, it was for the first time that statistical analysis and mathematical logic were applied to problems in biology. His experiment had a large sampling size which gave greater credibility to that data that he collected. Also, the confirmation of his inferences from experiments on successive generations of his test plants prove that his results pointed to general rules of inheritance rather than being unsubstantiated ideas. Mendel investigated characters in the garden pea plant that were manifested as two opposing traits, that is, tall or dwarf plants, yellow or green seeds. This allowed him to set up a basic framework of rules governing inheritance, which was explained on by later scientists to account for all the diverse natural observations and the complexity inherent in them. Mendel conducted such artificial pollination or cross-pollination experiments using several true breeding pea lines. A true breeding line is one that, having undergone continuous self-pollination, shows the stable trait inheritance and expression for several generations. Mendel selected 14 true breeding pea plant varieties as pairs which were similar except for one character with contrasting traits. Some of the contrasting traits selected were smooth or wrinkled seeds, yellow or green seeds, smooth or inflated pods, green or yellow pods, and tall or dwarf plants in figure 5.1 and table 5.1. 5.2 Inheritance of one gene. Let us take the example of one such hybridization experiment carried out by Mendel, where he crossed tall and dwarf pea plant to study the inheritance of one gene. He collected the seeds produced as a result of this cross and grew them to generate plants of the first hybrid generation. This generation is also called the filial progeny or the F1. Mendel observed that all the F1 progeny plants were tall. Like one of its parents, none were dwarf. He made similar observation for the other pairs of trait. He found that the F1 always resembled either one of the parents and that the trait of the other parent was not seen in them. Mendel then self-pollinated the tall F1 plants and to his surprise found that in the filial 2 generation, some of the offspring were dwarf. The character that was not seen in the F1 generation was now expressed. The proportion of plants that were dwarf were 1/4 of the F2 plant while 3/4 of the F2 plant were tall. The tall and dwarf traits were identical to their parental type and didn't show any blending. That is, all the offspring were either tall or dwarf, none were of in-between height. Similar results were obtained with the other traits that he studied. Only one of the parental traits was expressed in the F1 generation, while at the F2 stage, both the traits were expressed in the proportion 3:1. The contrasting traits did not show any blending at either F1 or F2 stage. Based on these observations, Mendel proposed that something was being stably passed down, unchanged from parent to offspring through the gametes over successive generations. He called these things as factors. Nowadays, we call them as genes. Genes, therefore, are the units of inheritance. They contain the information that is required to express a particular trait in an organism. Genes which code for a pair of contrasting traits are known as alleles. That is, they are slightly different forms of the same gene. If we use alphabetical symbols for each gene, then the capital letter is used for the trait expressed at the F1 stage and the small alphabet for the other trait. For example, in case of the character of height, capital T is used for the tall trait and small t for the dwarf, and capital T and small t are alleles of each other. Hence, in plants, the pair of alleles for height would be capital T, comma, capital T, small t, or small t. Mendel also proposed that in a true breeding, tall or dwarf pea variety, the allelic pair of genes for height are identical or homozygous. Capital T and small t respectively. Capital T and small t are called the genotype of the plant, while the descriptive terms tall and dwarf are the phenotype. What then would be the phenotype of a plant that had a genotype capital T and small t? As Mendel found the phenotype of the F1 heterozygote capital T small t to be exactly like the capital T parent in appearance, he proposed that in a pair of dissimilar factors, one dominates the other and hence is called the dominant factor, while the other factor is recessive. In this case, capital T is dominant over small t, that is recessive. He observed identical behavior for all the other characters or trait pair that he studied. It is convenient to use the capital N lowercase of an alphabetical symbol to remember this concept of dominance and recessiveness. Alleles can be similar as in the case of homozygotes capital T and small t, or can be dissimilar as in the case of heterozygote capital T and small t. Since the capital T small t plant is heterozygous for genes controlling one character, it is a monohybrid, and the cross between capital T and small t is a monohybrid cross. From the observation that the recessive parental trait is expressed without any blending in the F2 generation, we can infer that when the tall and dwarf plant produce gametes, by the process of meiosis, the alleles of the parental pair separate or segregate from each other and only one allele is transmitted to a gamete. This segregation of alleles is a random process, and so there is a 50% chances of a gamete containing either allele, as has been verified by the results of the crossings. In this way, the gametes of the tall capital T plants have the allele capital T, and the gametes of the dwarf plants have the allele small t. During fertilization, the two alleles capital T from one parent, say, through the pollen, and small t from the other parent, then through the egg, are united to produce zygotes that have one capital T allele and one small t allele. In other words, the hybrids have capital T small t. Since these hybrids contain alleles which express contrasting traits, the plants are heterozygous. The production of gametes by the parents, the formation of the zygotes, the F1 and F2 plants can be understood from a diagram called Punnett square as shown in the figure 5.4. It was developed by a British genetics Reginald C. Punnett. It is a graphical representation to calculate the probability of all possible genotypes of offspring in a genetic cross. The possible gametes are written on the two sides, usually the top row and left columns. All possible combinations are represented in boxes below in the squares, which generate a square output form. The Punnett square shows the parental tall and dwarf plants, the gametes produced by them, and the F1 capital T small t progeny. The F1 plants of genotype capital T small t are self-pollinated. The symbol male and female are used to denote the female and male of the F1 generation respectively. The F1 plant of the genotype capital T small t, when self-pollinated, produces gametes of the genotype capital T and small t in equal proportion. When fertilization takes place, the pollen grains of genotype capital T have a 50% chance to pollinate eggs of the genotype capital T as well as of genotype small t. Also, pollen grains of genotype small t have a 50% chance of pollinating eggs of genotype capital T as well as of genotype small t. As a result of random fertilization, the resultant zygotes can be of the genotype capital T, comma, capital T small t or small t small t. From the Punnett square, it is easily seen that 1/4 of the random fertilizations lead to capital T. 1/2 lead to capital T small t and 1/4 to small t. Though the F1 have a genotype of capital T small t, but the phenotypic character seen is tall. At F2, 3/4 of this plant are tall, where some of them are capital T while others are capital T small t. Externally, it is not possible to distinguish between the plants with the genotypes capital T and capital T small t. Hence, within the genotypic pair capital T small t, only one character capital T tall is expressed. Hence, the character capital T or tall is said to dominance over the other allele small t or dwarf character. It is thus due to this dominance of one character over the other that all the F1 are tall and the F2, 3/4 of the plants are tall. This leads to a phenotypic ratio of 3/4 tall. And 1/4 small t, that is a 3:1 ratio, but a genotypic ratio of 1:2:1. The 1/4 is to 1/2 is to 1/4 ratio of capital T small t is to small t is mathematically condensable to the form of the binomial expression in bracket a plus x plus b by whole square, that has the gametes bearing genes capital T or small t in equal frequency of 1/2. The expression is expanded as given below. Mendel self-pollinated the F2 plants and found that dwarf F2 plants continue to generate dwarf plants in F3 and F4 generations. He concluded that the genotype of the dwarf was homozygous small t. What do you think he would give God had self-pollinated a tall F2 plant? From the preceding paragraphs, it is clear that though the genotypic ratio can be calculated using mathematical probability by simply looking at the phenotype of a dominant trait, it is not possible to know the genotypic composition. That is, for example, whether a tall plant from F1 or F2 has capital T or capital T small t composition cannot be predicted. Therefore, to determine the genotype of a tall plant at F2, Mendel crossed the tall plant from F2 with a dwarf plant. This he called a taste cross. In a typical taste cross, an organism showing a dominant phenotype is crossed with a recessive parent instead of self-crossing. The progenies of such a cross can easily be analyzed to predict the genotype of the taste organism. Figure 5.5 shows the result of typical taste cross where violet color flower is dominant over the white color flower. Using Punnett square, try to find out the nature of offspring of a taste cross. What ratio did you get? Using the genotype of this cross, can you give a general definition for a taste cross? Based on his observation on monohybrid crosses, Mendel proposed two general rules to consolidate his understanding of inheritance in monohybrid crosses. Today, these rules are called the principles or laws of inheritance. The first law or law of dominance and the second law or law of segregation. 5.2.1 Law of Dominance. Characters are controlled by discrete units called factors. Factors occur in pairs. In a dissimilar pairs of factors, one member of the pair dominates the other. The law of dominance is used to explain the expression of only one of the parental characters in a monohybrid cross in the F1 and the expression of both in the F2. It also explains the proportion of 3:1 obtained at the F2. 5.2.2 Law of Segregation. This law is based on the fact that the alleles do not show any blending and that both the characters are recovered as such in the F2 generation, though one of this is not seen at the F1 stage. Though the parents contain two alleles during gamete formation, the factors or alleles of a pair segregate from each other such that a gamete receives only one of the two factors. Of course, a homozygous parent produces all gametes that are similar, while a heterozygous one produces two kinds of gametes, each having one allele with equal proportion. 5.2.2.1 Incomplete Dominance. When experiments on peas were repeated using other traits in other plants, it was found that sometimes the F1 had a phenotype that did not resemble either of the two parents and was in between the two. The inheritance of flower color in the dog flower is a good example to understand incomplete dominance. In a cross between true breeding red flower and true breeding white flowered plant, the F1 was pink. When the F1 was self-pollinated, the F2 resulted in the following ratio: 1 red is to 2 pink is to 1 white. Here the genotype ratio were exactly as we could expect in any Mendelian monohybrid cross, but the phenotype ratio had changed from the 3:1 dominant is to recessive ratio. 3:1 dominant is to recessive ratio. What happened was that R was not completely dominant over small r, and this made it possible to distinguish capital R small r as pink from capital R red and small r white. Explanation of the concept of dominance. What exactly is dominance? Why are some alleles dominant and some recessive? To tackle these questions, we must understand what a gene does. Every gene, as you know by now, contains the information to express a particular trait. In a diploid organism, there are two copies of each gene, that is, as a pair of alleles. Now, these two alleles need not always be identical, as in a heterozygote. One of them may be different due to some changes that it has undergone, which modify the information that particular allele contains. Let us take an example of a gene that contains the information for producing an enzyme. Now, there are two copies of this gene, the two allelic forms. Let us assume that the normal allele produces a normal enzyme that is needed for the transformation of a substrate S. Theoretically, the modified allele could be responsible for production of, first, the normal or less efficient enzyme, second, a non-functional enzyme, or third, no enzyme at all. In the first case, the modified allele is equivalent to the unmodified allele, that is, it will produce the same phenotype or trait, that is, result in the transformation of substrate S. Such equivalent allele pairs are very common, but if the allele produces a non-functional enzyme or no enzyme, the phenotype may be affected. The phenotype or trait will only be dependent on the functioning of the unmodified allele. The unmodified allele, which represent the original phenotype, is the dominant allele, and the modified allele is generally the recessive allele. Hence, in the example above, the recessive trait is seen due to non-functional enzyme or because no enzyme is produced. 5.2.2.2 Codominance. Till now, we were discussing crosses where the F1 resemble either of the two parents or was in between. But in the case of codominance, the F1 generation resembles both parents. A good example is different types of red blood cells that determine ABO blood grouping in human beings. ABO blood groups are controlled by the gene I. The plasma membrane of the red blood cells has sugar polymers that protrude from its surface, and the kind of sugar is controlled by the gene. The gene has three alleles, IA, IB and small I. The alleles IA and IB produces are slightly different from the sugar, while allele small I does not produce any sugar. Because humans are diploid organism, each person possesses any two of the three I gene alleles. IA and IB are completely dominant over small I. In the other word, when IA and small I are present, only IA expresses. And when IB and small I are present, IB expresses. But when IA and IB are present together, they both express their own types of sugars. This is because of codominance. Hence, RBC have both A and B types of sugar. Since there are three different alleles, there are 6 different combinations of these 3 alleles that are possible, a total of 6 different genotypes of the human ABO blood types. How many phenotypes are possible? Do you realize that the example of ABO blood grouping also provides a good example of multiple alleles? Here you can see that there are more than 2, that is 3 alleles governing the same character. Since in an individual only 2 alleles can be present, multiple alleles can be found only when population studies are made. Occasionally, a single gene product may produce more than one effect. For example, starch synthesis in pea seeds is controlled by one gene. It is 2 alleles. Starch is synthesized effectively by capital B, homozygotes, and therefore large starch grains are produced. In contrast, small b, homozygotes have lesser efficiency in starch synthesis and produce smaller starch grains. After maturation of the seeds, capital B seeds are round and the small b seeds are wrinkled. Heterozygotes produce round seeds, and so capital B seems to be dominant allele. But the starch grain produces are of intermediate size in capital B small b seeds. So if the starch grain size is considered as the phenotype, then from the angle the alleles show incomplete dominance. Therefore, dominance is not an autonomous feature of a gene or the product that is it has information for. It depends as much as the gene product and the production of a particular phenotype from this product as it does on the particular phenotype that we choose to examine in case more than one phenotype is influenced by the same gene. 5.3 Inheritance of two genes. Mendel also worked with and crossed pea plants that differ in 2 characters, as is seen in the cross between a pea plant that has seeds with yellow color and round shape and one that had seeds of green color and wrinkled shape. Mendel found that the seeds resulting from the crossing of the parents had yellow colored and rounded shaped seeds. Here, can you tell which of the characters in the pair's yellow or green color and round or wrinkled shape was dominant? Thus, yellow color was dominant over green and round shape dominant over wrinkle. These results were identical to those that he got when he made separate monohybrid crosses between yellow and green seeded plants and between round and wrinkled seed plants. Let us use the genotypic symbols capital Y for dominant yellow seed color and small y for recessive green seed color, capital R for round shaped seeds and small r for wrinkled seed. The genotype of the parents can be written as capital R, capital Y, and small r, small y. The cross between the 2 plants can be written down as in figure 5.7 showing the genotypes of the parent plants. The gametes capital R, capital Y and small y unite on fertilization to produce the F1 hybrid capital R, capital Y, small y. When Mendel self-hybridized the F1 plants, he found that 3/4 of F2 plants had yellow seeds and 1/4 had green. The yellow and green color segregated in a 3:1 ratio. Round and wrinkled seed shape also segregated in a 3:1 ratio, just like in a monohybrid cross. 5.3.1 Law of Independent Assortment. In the dihybrid cross, the phenotypes round yellow, wrinkled yellow, round green and wrinkled green appeared in the ratio 9:3:3:1. Such a ratio was observed for several pairs of characters that Mendel studied. The ratio of 9:3:3:1 can be derived as a combination series of 3 yellow, 1 green with 3 round and 1 wrinkle. This derivation can be written as follows. 3 round is to 1 wrinkle, 3 yellow is to 1 green is equals to 9 round, yellow is to 3 wrinkle, yellow is to 3 round, green is to 1 wrinkle, green. Based upon such observations on dihybrid crosses, Mendel proposed a second set of generalization that we call Mendel's law of independent assortment. The law states that when two pairs of traits are combined in a hybrid, segregation of one pair of characters is independent of the other pair of characters. The Punnett square can be effectively used to understand the independent segregation of the two pairs of genes during meiosis and the production of eggs and pollen in the F1, capital R, small y plant. Consider the segregation of one pair of genes capital R and small r. 50% of the gametes have the gene capital R and the other 50% have small r. Now besides each gamete having either capital R or small r, it should also have the allele capital Y or small y. The important thing to remember here is that segregation of 50% capital R and 50% small r is independent from the segregation of 50% capital Y and 50% small y. Therefore, 50% of the small r bearing gametes has capital Y and the other 50% has small y. Similarly, 50% of the capital R bearing gametes has capital Y and the other 50% has small y. Thus, there are 4 genotypes of gametes. The 4 types are capital R capital Y, capital R small y, small r capital Y and small r small y, each with a frequency of 25% or 1/4 of the total gametes produced. When you write down the 4 types of eggs and pollen on the two sides of a Punnett square, it is very easy to derive the composition of the zygotes that give rise to the F2 plants. Although there are 16 squares, how many different types of genotype and phenotype are formed? Note them down in the format given. Can you, using the Punnett square data, work out the genotypic ratio at the F2 stage and fill the format given? Is the genotypic ratio also 9:3:3:1? 5.3.2 Chromosomal Theory of Inheritance. Mendel published his work on inheritance of characters in 1865, but for several reasons it remained unrecognized till 1900. Firstly, communication was not easy in those days and his work could not be widely published. Secondly, his concept of genes as stable and discrete units that control the expression of traits and of the pair of alleles which did not blend with each other was not accepted by his contemporaries as an explanation for the apparently continuous variations seen in nature. Thirdly, Mendel's approach of using mathematics to explain biological phenomena was totally new and unacceptable to many of the biologists of his time. Finally, though Mendel's work suggested that factors were discrete units, he could not provide any physical proof for the existence of factors or say what they were made of. In 1900, 3 scientists, D. Rice, Correns and von Sarmac, independently rediscovered Mendel's results on the inheritance of characters. Also by this time, due to advancement in microscopy they were taking place, scientists were able to carefully observe cell division. This led to the discovery of structures in the nucleus that appear to double and divide just before each cell division. These were called chromosomes. By 1902, the chromosome movement during meiosis had been worked out. Walter Sutton and Theodore Boveri noted that the behavior of chromosomes was parallel to the behavior of genes and used chromosome movement to explain Mendel's laws. Recall that you have studied the behavior of chromosomes during mitosis and during meiosis. The important things to remember are that chromosomes as well as genes occur in pairs. The 2 alleles of a gene pair are located on homologous sites on homologous chromosomes. During anaphase of meiosis 1, the two chromosome pair can align at the metaphase plate independently of each other. To understand this, compare the chromosomes of 4 different colors in the left and right column. In the left column, orange and gene is segregating together. But in the right hand column, the orange chromosome is segregating with the red chromosomes. Possibility 1. One long orange and short green chromosome and long yellow and short red chromosome at the same pole. Possibility 2. One long orange and short red chromosome and long yellow and short green chromosome at the same pole. Sutton and Boveri argue that the pairing and separation of a pair of chromosomes would lead to the segregation of a pair of factors they carried. Sutton united the knowledge of chromosomal segregation with Mendelian principles and called it the chromosomal theory of inheritance. Following this synthesis of ideas, experimental verification of the chromosomal theory of inheritance by Thomas Hunt Morgan and his colleagues led to discovering the basis for the variation that sexual reproduction produced. Morgan worked with the tiny fruit flies Drosophila melanogaster, which were found very suitable for such studies. They could be grown on simple synthetic medium in the laboratory. They complete their life cycle in about 2 weeks, and a single mating could produce a large number of progeny flies. Also, there was a clear differentiation of the sexes. The male and female flies are easily distinguishable. Also, it has many types of hereditary variations that can be seen with low power microscopes. 5.3.3 Linkage and Recombination. Morgan carried out several dihybrid crosses in Drosophila to study genes that were sex-linked. The crosses were similar to the dihybrid crosses carried out by Mendelian peas. For example, Morgan hybridized yellow body, white-eyed females to brown body, red-eyed males, and intercrossed their F1 progeny. He observed that the two genes did not segregate independently of each other, and the F2 ratio deviated very significantly from the 9:3:3:1 ratio. Morgan and his group knew that the genes were located on the X chromosome and showed quickly that when the two genes in a dihybrid cross were situated on the same chromosome, the proportion of parental gene combinations were much higher than the non-parental type. Morgan attributed this due to the physical association or linkage of the two genes and coined the term linkage to describe this physical association of genes on a chromosome, and the term recombination to describe the generation of non-parental gene combinations. Morgan and his group also found that even when genes were grouped on the same chromosome, some genes were very tightly linked, while others were loosely linked. For example, he found that the genes white and yellow were very tightly linked and show only 1.3% recombination, while white and miniature wing showed 37.2% recombination. His student Alfred Sturtevant used this frequency of recombination between gene pair on the same chromosome as a measure of the distance between genes and mapped their position on the chromosome. Today, genetic maps are extensively used as a starting point in the sequencing of whole genomes, as was done in the case of the Human Genome Sequencing Project. Describe later. (5.4) Sex determination. The mechanism of sex determination has always been a puzzle before the genetics. The initial clue about the genetic or chromosomal mechanism of sex determination can be traced back to some of the experiments carried out in insects. In fact, the cytological observations made in a number of insects led to the development of the concept of genetic or chromosomal basis of sex determination. Hanking could trace a specific nuclear structure all through spermatogenesis in a few insects, and it was also observed by him that 50% of the sperm received this structure after spermatogenesis, whereas the other 50% sperm did not receive it. Hanking gave a name to the structure as X-body, but he could not explain its significance. Further investigations by other scientists led to the conclusion that the X-body of Hanking was in fact a chromosome, and that is why it was given the name X chromosome. It was also observed that in a large number of insects, the mechanism of sex determination is of the X0 type, that is, all eggs bear an additional X chromosome besides the other chromosome. On the other hand, some of the sperms bear the X chromosome, whereas some do not. Eggs fertilized by sperm having an X chromosome become females, and those fertilized by sperm that do not have an X chromosome become male. Do you think the number of chromosomes in the male and female are equal? Due to the involvement of the X chromosome in the determination of sex, it was designated to by the sex chromosome, and the rest of the chromosomes were named as autosomes. Grasshopper is an example of X0 type of sex determination in which the males have only one X chromosome besides the autosomes, whereas females have a pair of X chromosomes. These observations led to the investigation of a number of species to understand the mechanism of sex determination. In a number of other insects and mammals, including man, XY type of sex determination is seen where both male and female have same number of chromosomes. Among the males, an X chromosome is present, but its counterpart is distinctly smaller and called the Y chromosome. Females, however, have a pair of X chromosomes. Both males and females bear same number of autosomes. Hence, the males have autosomes plus XY, while female have autosomes plus XX. In human beings and in Drosophila, the males have one X and one Y chromosome, whereas females have a pair of X chromosomes beside autosomes. In the above description, you have studied about two types of sex determining mechanisms, that is X0 type and XY type. But in both cases, males produce two different types of gametes. A, either with or without X chromosome, or B, some gametes with X chromosome and some with Y chromosome. Such types of sex determination mechanism is designated to be the example of male heterogamety. In some other organism, that is birds, a different mechanism of sex determination is observed. In this case, the total number of chromosomes is same in both males and females, but two different types of gametes in terms of the sex chromosomes are produced by females, that is female heterogamety. In order to have a distinction with the mechanism of sex determination described earlier, the two different sex chromosomes of a female bird has been designated to be the Z and W chromosomes. In this organism, the female have one Z and one W chromosome, whereas males have a pair of Z chromosomes besides the autosomes. 5.4.1 Sex determination in humans. It has already been mentioned that the sex determining mechanism in case of humans is XY type. Out of 23 pairs of chromosomes present, 22 pairs are exactly same in both males and females. These are the autosomes. A pair of X chromosome are present in the female, whereas the presence of an X and Y chromosome are determinant of the male characteristic. During spermatogenesis, among males, two types of gametes are produced. 50% of the total sperm produced carry the X chromosome, and the rest 50% has Y chromosome besides the autosomes. Females, however, produce only one type of ovum with an X chromosome. There is an equal probability of fertilization of the ovum with the sperm carrying either X or Y chromosome. In case the ovum fertilizes with a sperm carrying X chromosome, the zygote develops into a female, and the fertilization of ovum with Y chromosome carrying sperm results into a male offspring. Thus, it is evident that it is the genetic makeup of the sperm that determines the sex of the child. It is also evident that in each pregnancy, there is always 50% probability of either a male or a female child. It is unfortunate that in our society, women are blamed for producing female children and have been ostracized and ill-treated because of this false notion. How is the sex determination mechanism different in the birds? Is the sperm or the egg responsible for the sex of the chicks? 5.5 Mutation. Mutation is a phenomenon which results in alteration of DNA sequences and consequently results in changes in the genotype and the phenotype of an organism. In addition to recombination, mutation is another phenomenon that leads to variation in DNA. As you will learn in chapter 6, one DNA helix runs continuously from one end to the other in each chromatid in a highly supercoiled form. Therefore, loss or gain of a segment of DNA result in alteration in chromosomes. Since genes are known to be located on chromosomes, alteration in chromosomes result in abnormalities or aberrations. Chromosomal aberrations are commonly observed in cancer cells. In addition to the above, mutation also arises due to change in a single base pair of DNA. This is known as point mutation. A classical example of such a mutation is sickle cell anemia. Deletions and insertions of base pairs of DNA causes frame shift mutations. The mechanism of mutation is beyond the scope of this discussion at this level. However, there are many chemical and physical factors that induce mutations. These are referred to as mutagens. UV radiations can cause mutations in organisms. It is a mutagen. 5.6 Genetic disorders. 5.6.1 Pedigree analysis. The idea that disorders are inherited has been prevailing in the human society since long. This was based on the heritability of certain characteristic features in families. After the rediscovery of Mendel's work, the practice of analyzing inheritance pattern of traits in human beings began. Since it is evident that control crosses that can be performed in pea plant or some other organisms are not possible in case of human beings, study of the family history about inheritance of a particular trait provides an alternative. Such an analysis of traits in several of generations of a family is called the pedigree analysis. In the pedigree analysis, the inheritance of a particular trait is represented in the family tree over generations. In human genetics, pedigree study provides a strong tool which is utilized to trace the inheritance of a specific trait, abnormality or disease. Some of the important standard symbols used in the pedigree analysis have been shown in figure 5.13. As you have studied in this chapter, each and every feature in any organism is controlled by one or other gene located on the DNA present in the chromosome. DNA is the carrier of genetic information. It is hence transmitted from one generation to the other without any change or alteration. However, changes or alteration do take place occasionally. Such an alteration or change in the genetic material is referred to as mutation. A number of disorders in human beings have been found to be associated with the inheritance of change or altered genes or chromosomes. 5.6.2 Mendelian disorders. Broadly, genetic disorders may be grouped into two categories: Mendelian disorders and chromosomal disorders. Mendelian disorders are mainly determined by alteration or mutation in the single gene. These disorders are transmitted to the offspring on the same lines as we have studied in the principle of inheritance. The pattern of inheritance of such Mendelian disorders can be traced in a family by the pedigree analysis. Most common and prevalent Mendelian disorders are hemophilia, cystic fibrosis, sickle cell anemia, color blindness, phenylketonuria, thalassemia, etc. It is important to mention here that such Mendelian disorders may be dominant or recessive. By pedigree analysis, one can easily understand whether the trait in question is dominant or recessive. Similarly, the trait may also be linked to the sex chromosome as in case of hemophilia. It is evident that this X-linked recessive trait shows transmission from carrier female to male progeny. A representative pedigree is shown in figure 5.14 for dominant and recessive trait. Discuss with your teacher and design pedigrees for characters linked to both autosomes and sex chromosome. Hemophilia. This sex-linked recessive disease will show its transmission from unaffected carrier female to some of the male progeny has been widely studied. In this disease, a single protein that is a part of the cascade of proteins involved in the clotting of blood is affected. Due to this, in an affected individual, a simple cut will result in non-stop bleeding. The heterozygous female for hemophilia may transmit the disease to sons. The possibility of a female becoming a hemophilic is extremely rare because mother of such a female has to be a least carrier and the father should be hemophilic. The family pedigree of Queen Victoria shows a number of hemophilic descendants as she was a carrier of the disease. Sickle cell anemia. This is an autosome-linked recessive trait that can be transmitted from parents to the offspring when both the parents are carrier for the gene. The disease is controlled by a single pair of allele, HBA and HBS. Out of the 3 possible genotypes, only homozygous individual for HBS show the disease phenotype. Heterozygous individual appear apparently unaffected, but they are carrier of the disease as there is 50% probability of transmission of the mutant gene to the progeny, thus exhibiting sickle cell trait. The defect is caused by substitution of glutamic acid by valine at the 6th position of the beta globin chain of the hemoglobin molecule. The substitution of amino acid in the globin protein result due to the single base substitution at the 6th codon of the beta globin gene from GAG to GUG. The mutant hemoglobin molecule undergoes polymerization under low oxygen tension, causing the change in the shape of the RBC from biconcave disc to elongated sickle-like structure. Phenylketonuria. This inborn error of metabolism is also inherited as the autosomal recessive trait. The affected individual lacks an enzyme that converts the amino acid phenylalanine into tyrosine. As a result of this, phenylalanine is accumulated and converted into phenylpyruvic acid and other derivatives. Accumulation of this in brain results in mental retardation. These are also excreted through urine because of its poor absorption by kidney. 5.6.3 Chromosomal disorders. The chromosomal disorders, on the other hand, are caused due to the absence or excess or abnormal arrangement of one or more chromosomes. Failure of segregation of chromatids during cell division cycle results in the gain or loss of chromosomes called aneuploidy. For example, Down syndrome results in the gain or extra copy of chromosome 21. Similarly, Turner syndrome results due to the loss of an X chromosome in human females. Failure of cytokinesis after telophase stage of cell division results in an increase in a whole set of chromosomes in an organism, and this phenomenon is known as polyploidy. This condition is often seen in plants. The total number of chromosomes in a normal human is 46, in bracket 23 pairs. Out of this, 22 pairs are autosomes and 1 pair of chromosomes are sex chromosomes. Sometimes, though rarely, either an additional copy of a chromosome may be included in an individual, or an individual may lack one or any one pair of chromosomes. These situations are known as trisomy or monosomy of a chromosome respectively. Such a situation leads to very serious consequences in the individual. Down syndrome, Turner syndrome, Klinefelter syndrome are common examples of chromosomal disorders. Down syndrome. The cause of this genetic disorder is the presence of an additional copy of the chromosome number 21, in bracket trisomy of 21. This disorder was first described by Langdon Down in 1866. The affected individual is short-statured with small round head, furrowed tongue and partially open mouth. Palm is broad with characteristic palm crease. Physical, psychomotor and mental development is retarded. Klinefelter syndrome. This genetic disorder is also caused due to the presence of an additional copy of X chromosome resulting into a karyotype of 47 XXY. Such an individual has overall masculine development. However, the feminine development, in bracket development of breast, gynecomastia, is also expressed. Such individuals are sterile. Turner syndrome. Such a disorder is caused due to the absence of one of the X chromosome, that is 45 with X0. Such females are sterile as ovaries are rudimentary besides other features including lack of the other secondary sexual characters.